Hello everyone and welcome to the new session of the lecture series, and today's topic is respiration in plants. So this lecture is completely NCERT based, means all the topics covered in this lecture from the NCERT book, and this lecture is the continuation of the previous lecture. So let me again introduce you the term respiration. Alright, so respiration is a metabolic process, that's involved breaking of the carbon-carbon bonds of the complex organic molecules. Alright, these complex organic molecules are known as the respiratory substrate. Alright, so these respiratory substrate or the complex organic compounds are broken down, with the help of oxygen. So it is an oxidation reaction, and what happens? Finally, there is a release of considerable amount of energy in the form of ATP. So respiration is basically conversion of complex organic compound into simpler organic compounds, and in the process, energy is released in the form of ATP. Now in the previous lecture, we were discussing about the glycolysis pathway. Alright, now we already know that glycolysis, the first phase of glycolysis is the preparatory phase. First phase is known as the preparatory phase, during which ATP is consumed. Instead of synthesizing ATP, ATP is consumed during the preparatory phase. So till here, till the fifth stage, this is known as the preparatory phase, or you can say till the formation of glyceraldehyde 3-phosphate. This is known as the preparatory phase. Alright, so this is the breakdown of the glycolysis pathway, which is also known as Embden-Meyerhof-Parnas pathway. Alright, so let's discuss about glycolysis in detail now. Alright, so this is the preparatory phase. This is the preparatory phase. Till here, till the synthesis of glyceraldehyde 3-phosphate. So from glucose, which is a 6-carbon compound, a 3-carbon compound is produced. And 2 molecules of glyceraldehyde 3-phosphate is produced. Alright, now we have already discussed the classes of enzymes. Kinases are involved, mutases are involved. Then we have aldolase here. Alright, and again we have isomerase. In the preparatory phase. So now, when the glucose is converted into glucose 6-phosphate, this is the first step of glycolysis. This is the phosphorylation step, because phosphate is being added to the glucose molecule. Alright, so first step is known as phosphorylation. First step is phosphorylation, where phosphate is being added and ATP is consumed and ADP is formed. Alright, this reaction is catalyzed by the enzyme hexokinase. Now the next step is step 2. Glucose is isomerized into fructose 6-phosphate. So the second step is isomerization. Second step is the isomerization, which is catalyzed by phosphoglucomutase, or you can also say phosphoglucoisomerase. Alright, what is the role of mutase? It do the molecular rearrangement. Alright, and especially of the phosphate group, which is attached to the hydroxyl group. Alright, so this is the isomerization step. Now third step is again the phosphorylation step. So third step, again what is happening? Phosphorylation is happening, addition of phosphate is taking place. So this is the second phosphorylation step. second phosphorylation is taking place here. Step 1 was the first phosphorylation. Okay. So this reaction is catalyzed by the enzyme phosphofructokinase. And an ATP is consumed in this process. Now next step, which is the fourth step, okay, in this step what happens? Fructose 1,6-bisphosphate is converted into one molecule of dihydroxyacetone phosphate and one molecule of glyceraldehyde 3-phosphate. Both of these are 3-carbon compound. Alright, so this step is known as the cleavage step. Okay, this step is known as the cleavage step, and this is catalyzed by the enzyme aldolase. Alright, so aldolase is a lyase enzyme. Okay, it is a lyase enzyme, means what it do? It breaks the chemical bonds of the organic molecules. So here, with the activity of aldolase, fructose 1,6-bisphosphate is converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Alright. Now, what happens? The dihydroxyacetone phosphate, okay, is further converted into glyceraldehyde 3-phosphate. Alright. So in the end, we have 2 molecules of glyceraldehyde 3-phosphate. And this reaction is catalyzed by the enzyme triose phosphate isomerase. Okay. Why we call it triose? What is a triose? Triose is a compound which consists of 3 carbons. Okay. That's why triose. And it contains a phosphate group. That's why triose phosphate. So at the end of preparatory phase, what we have seen, there is formation of 2 molecules of glyceraldehyde 3-phosphate. Okay, which is a 3-carbon compound. And what else we have seen? 2 ATP are utilized here or consumed, okay, in the 2 phosphorylation steps. Okay. Now next come to the next step, which is the 6th step. And what is happening here? Oxidation of glyceraldehyde 3-phosphate takes place into 1,3-bisphosphoglycerate. Okay, or you can also write this as glyceric acid. Okay, 1,3-bisphosphoglyceric acid. Alright, so this step, step 6, is known as the oxidation step. This is known as the oxidation step, and this reaction is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Okay, so you can see another class of enzyme dehydrogenase are involved here. And what is the function of dehydrogenase? Dehydrogenate catalyze the oxidation reduction reaction. Alright, so what does dehydrogenase do? They catalyze the, you can write oxidoreductase oxidoreduction reactions. Okay, means it transfers the electrons from the electron donor to the electron acceptor. Alright, what is oxidation? Oxidation is loss of electrons, whereas reduction is gain of electrons. Now one thing to note here is that NAD, okay, which is a coenzyme, is converted into NADH. Alright, another thing to note here is that NADH is being produced. Alright, and 2 molecules of NADH are produced because here we have 2 molecules of glyceraldehyde 3-phosphate. Alright, so from here now From here on, everything will be, okay, twice. Okay. So now the next step. In the next step, which is the step 7, what is happening? 1,3-bisphosphoglyceric acid or glycerate is converted into 3-phosphoglyceric acid. So what is happening here? This step is known as the dephosphorylation step. Seventh step is, this is the first dephosphorylation. Means there is loss of the phosphate. Okay, you can see there is one less phosphate here. And what happens? This phosphate is accepted by adenosine diphosphate. Okay, and ADP is converted into adenosine triphosphate, ATP. Okay, so again, 2 molecules of ATP are generated here. Alright. So now coming to the next stage, or next step, which is the 8th step. In this, what is happening? 3-phosphoglycerate is converted into 2-phosphoglycerate. Alright. So this reaction is again catalyzed by the enzyme phosphoglyceromutase. Alright. Now, this is again a kind of isomerization. So you can write here as isomerization. Now next step, which is the 9th step, okay. Here what happens, 2-phosphoglycerate is converted into phosphoenol pyruvate. Okay, so this step is known as the dehydration step. This is known as the dehydration step. Okay, and this reaction is catalyzed by the enzyme enolase. And what happens? There is a release of water molecule here. That's why it is known as the dehydration step. Now, what is the role of enolase? Enolase helps in the removal of hydroxyl group. Alright. And this leads to the release of water molecules. 2 molecules of water are released. Now finally, let's talk about the 10th step, okay, which is the final step. Here what happens? Phosphoenol pyruvate, okay, phosphoenol pyruvate is converted into pyruvate or pyruvic acid. Pyruvic acid can also be called as pyruvate. Now here, what is happening? Dephosphorylation is again happening here. This is the second dephosphorylation step. Okay, you can see here, ADP accepts the phosphate and is converted into ATP. Okay. So now you can see glycolysis is a 10-step process, or glycolysis consists of 10 steps and it also contains 10 enzymes. Okay, so let's see what is the outcome of the glycolysis now. Okay, first you have to notice that what is happening, NAD is being synthesized here. Alright, in the step 6, alright, NAD was synthesized in the step 6. This step, NAD is synthesized here. And what is happening? Later on, ATP is also synthesized. Okay, you have to remember these steps. 6th step you have to remember, 7th step you have to remember. Okay, you get questions from them, these steps. Okay, where is ATP synthesized? At which step is ATP synthesized? At which step is NADH is synthesized? Alright, so now let's talk about the net outcome or the net gain of glycolysis. Okay, so now we know in the preparatory phase what happened. 2 molecules of ATP was consumed. Okay, But later on, what happens? 4 ATP was produced, okay, and as well as 2 NADH molecules are produced. So what is the net outcome? Net outcome will be 4 minus 2, which is 2 ATP. 2 ATP are produced along with 2 molecules of NADH. Alright. Now, alright, so this is the net gain or the net outcome of the glycolysis pathway. Now we know that 1 NADH molecule, okay, is equivalent to 3 ATP. Alright, so what will be the total ATP here? 3 into 2 is 6 ATP. 6 ATP plus 2 ATP will be 8 ATP. So 8 ATP are generated during the glycolysis pathway. Alright, so this should be clear. Alright, now moving on to the next step. Now pyruvic acid is or pyruvate is produced now. Alright, 2 molecules of pyruvates are produced. Now what will happen? Depending on the conditions or depending on the organism, pyruvic acid can either enter the aerobic respiration, alright, or it can follow the anaerobic pathway. Okay, in aerobic respiration, oxygen is required, whereas anaerobic respiration happens without the oxygen. Alright, so let's talk about aerobic respiration first. If the pyruvic acid enters the aerobic cycle, okay, then complete oxidation of pyruvic acid takes place, okay, and this happens with the help of Krebs cycle, okay. So, but if the pyruvic acid follows anaerobic pathway, okay, then incomplete oxidation of pyruvic acid takes place, or you can say incomplete oxidation of glucose takes place, okay, and this takes place with the help of alcoholic fermentation, and second one is lactic acid fermentation. So anaerobic respiration is also known as fermentation, and there are 2 types of fermentation: alcoholic fermentation and lactic acid fermentation. Now first let's talk about anaerobic respiration, okay, and within anaerobic respiration, first we will talk about the alcoholic fermentation. So what happens during alcoholic fermentation? Pyruvic acid, okay, or the pyruvate which is formed, okay, during glycolysis. And one thing again you have to remember that glycolysis is a common pathway here, okay, it is a common pathway before aerobic and anaerobic respiration. So you can write here glycolysis is an anaerobic pathway also, and it is a common pathway. Is a common pathway for both aerobic respiration as well as anaerobic respiration, and it leads to the production of pyruvate, 2 molecules of pyruvate, and pyruvate can either enter aerobic pathway or it can follow the anaerobic pathway. Now coming back to the alcoholic fermentation. So the pyruvate which is produced, okay, is finally converted into ethanol and carbon dioxide during alcoholic fermentation. Okay, so ethanol and carbon dioxide are produced as byproducts during alcoholic fermentation. And an example of organism where alcoholic fermentation is seen is yeast. Okay, what is yeast? Yeast is a unicellular. Okay, it is an exception. It is a unicellular eukaryote. Okay, and in the process, what happens during alcoholic fermentation? NADH, okay, is converted into NAD. Alright, During glycolysis, NADH was produced, but here what is happening? NADH is consumed and it is again converted back to NAD. Alright. So NADH is consumed. Okay, is consumed. Now what is NADH? NADH are, I've already told you in previous lecture, it is an coenzyme. What are coenzymes? Coenzymes are organic molecules, okay, that bind to the active site of enzymes. Okay, they bind to the active site of enzymes, okay, and is responsible for the catalytic reaction of the enzymes. Okay, so NADH is nicotinamide adenine diphosphate. Okay, so what happens during alcoholic fermentation? These are the final product, but what happens actually? If this is pyruvate, pyruvate is first converted into acetaldehyde. Okay, it is first converted into acetaldehyde. So this is a 3-carbon compound, whereas acetaldehyde is a 2-carbon compound. Alright, so here carbon dioxide is released at this step. And then later acetaldehyde is finally converted into ethanol, which is an alcohol. That's why it has been given the name alcoholic fermentation. Okay. Here you can see net gain I have written is 2 ATP only. Why? Because in glycolysis, what happened? 2 ATP were synthesized during the glycolysis. Okay, we've already seen the net outcome of glycolysis. 2 ATP were generated. Okay, but what is happening in alcoholic fermentation? 2 NADH are being utilized. So what should be the outcome? Outcome should be only 2 ATP. Now let's talk about the lactic acid fermentation. Alright, so lactic acid fermentation, okay, some of the examples first I will give. Some of the examples of the organism where lactic acid fermentation takes place are lactic acid bacteria, okay. So what are the examples of lactic acid bacteria? First is lactobacillus. Another you can write lactococcus. In both of them, lactic acid fermentation is seen. Okay, this pathway is observed in these bacteria. Alright. Now another example can be lactic acid fermentation can also takes place during the oxygen depleted condition or when there is lack of oxygen. So sometimes in animals, okay, or humans, you can say it takes place in muscles. Okay, when there is oxygen, lack of oxygen or there is oxygen depletion. So lactic acid is produced from pyruvic acid. Alright. And later on what happens? When there is oxygen rich condition, then lactic acid is again converted back to pyruvic acid. Okay, lactic acid, okay, goes to liver and is again converted back to pyruvic acid. So what is the significance of lactic acid fermentation? Alright. So it provides a survival strategy, okay, during intense exercise conditions, okay, like in humans, okay, example is muscles, or in oxygen depleted conditions. Okay, examples are some animals, okay, like humans, and bacteria such as lactic acid bacteria. Alright, so again what is happening here? Pyruvic acid is produced, okay, okay, during glycolysis, and the pyruvic acid is finally converted into lactic acid. Now again you can see here NADH. Okay, 2 molecules of NADH will be used because there is 2 molecules of pyruvic acid. Alright. Okay, so what should be the outcome of lactic acid also? It is again 2 ATP. Okay, 2 ATP are produced or generated during the anaerobic respiration, okay, or during the alcoholic fermentation or lactic acid fermentation. Okay, now next we will be talking about the aerobic respiration. I'll give you a brief idea about that today. Alright, so after glycolysis, okay, pyruvic acid was produced. Now, if pyruvic acid, okay, enters the aerobic pathway, so first, okay, it has to be converted into some molecule, okay, which is known as acetyl coenzyme A. Okay, so pyruvic acid doesn't enter directly into the respiratory pathway or the Krebs cycle. We'll talk about this cycle, okay, in the next lecture. Okay, so pyruvic acid is first converted into acetyl coenzyme A, and then acetyl coenzyme A will enter the Krebs cycle, which takes place in the mitochondria. Alright, so this conversion of pyruvic acid to acetyl coenzyme A, this is known as oxidative decarboxylation of pyruvic acid, okay, or you can say aerobic oxidation of pyruvic acid. Okay, decarboxylation, why we say it a decarboxylation? Because there is a loss of carbon dioxide. Okay, pyruvic acid loses a carbon dioxide molecule and forms acetyl coenzyme A. Okay, and why it is called oxidative? Okay, why it is called oxidative? Because due to the oxidation reactions, due to the oxidation, or you can say due to the oxidation reactions, the carboxyl group, okay, the carboxyl group is converted into carbon dioxide. Is converted into carbon dioxide. That's why it is known as oxidative decarboxylation. So here you can see pyruvic acid, okay, which was produced where? In the cytoplasm, because glycolysis takes place in cytoplasm or cytosol. So here glycolysis is taking place in cytoplasm of the cell. Here pyruvic acid is produced. Again 2 molecules of pyruvic acid is produced. Now with the help of a transport protein, okay, these are transport protein, pyruvic acid enters the mitochondria. Okay, so here they have shown mitochondria, and this is the matrix of mitochondria. Matrix means the fluid of the mitochondria. And what is the importance of matrix? Matrix contains all the enzymes required for the biochemical reaction. It contains all the enzymes. It also consists of coenzymes such as NAD and NADPH. NAD and NADP. NADH, okay. Or you can write NADP also. Alright. So now, coming back to this figure, here pyruvic acid enters the matrix. Alright, it enters the matrix, and what happens? Pyruvic acid loses a carbon dioxide first. Alright, first step is loss of carbon dioxide. Second is there is addition of coenzyme A. Alright, now what is coenzyme A? coenzyme A is a cofactor. Okay, and now what is a cofactor? Cofactor is a, you can say, metallic ion or non-protein. It is a non-protein compound, okay, or metallic ion that is important for the role of the catalysis of enzyme. Okay, without that the enzyme can lose its catalytic power. Okay, so these cofactor bind to the enzymes and is required for the catalysis of the enzyme. Okay, now third thing which is happening is NAD. Again, NAD is converted into NADH. Alright, so these three things are happening. This is very important. A lot of questions can come from this reaction. Okay, and finally acetyl coenzyme A is produced. Alright, which is a 2-carbon compound. So from 3-carbon compound, 2-carbon compound acetyl coenzyme A is produced. Alright, and again 2 molecules of pyruvic acid was produced, so 2 molecules of acetyl coenzyme will be produced, 2 molecules of carbon dioxide will be released, 2 molecules of coenzyme A will be added, okay, individually, and 2 molecules of NAD plus is converted into 2 molecules of NADH. Okay, so what will be the net outcome here? Okay, if we talk about the net outcome of this reaction or net gain, we can say we have 2 NADH are synthesized here. Okay, 2 carbon dioxide are lost and coenzyme A is added. So now 1 NADH we know is equivalent to 3 ATP, so means 6 ATP. This will be equivalent to 6 ATP. This also you have to remember. Alright. Now this acetyl coenzyme A produced will enter the Krebs cycle now. Alright. So this was the end of this session and we'll be discussing about the Krebs cycle later in detail in the next lecture. Thank you very much for listening.